The FDA Modernization Act 2.0 was really a landmark piece of legislation. I think it's changed the game overnight for labs like mine. And whereas before, we were very well established as discovery tools for drug development, now we're really at the end stages of those space. We're an IND enabling tool, and we are now thinking much more about the regulatory applications and utilizing these systems to keep people safer, to make the therapeutic dosing right so that the clinical trials can actually succeed instead of having to be reperformed. We're considering using cells from a person, from their own body, as an avatar for themselves in a clinical trial, and having that as a living piece of feedback during the trial. So there's all sorts of possibilities now that didn't exist before. And I do believe that we are on the road to at least reducing, if not fully replacing, the use of animal models in clinical trial design and regulatory processes.
I think the trend started prior to the FDA Modernization Act because of the great failure rate in most drugs. Again, only about 5% of drugs actually make it through the FDA process, and it's mostly due to two reasons: lack of efficacy as well as toxicity. And those aren't typically modeled very well either in 2D culture or animal models. So I think you started seeing more and more liver organoids for toxicity and putting things up earlier, I should say, in the drug discovery pathway. I think now, with the aspect that you don't need animal models for FDA approval, I think you'll start seeing more and more sophisticated models that can help mimic efficacy, toxicity, as well as biomarker analysis, especially in drug discovery.
Yeah, so I've just seen a huge increase in the number of peer-reviewed papers around three-dimensional cell culture, organoid models, and certainly I hear from more and more customers every day asking how to get started with these types of models. I think everybody sees the value in either moving away from animal-based models or at least supplementing those applications.
3D models and the Modernization Act have really changed, I think, the way we will approach testing in animals and validation of drug responses in animals. Over time, I believe it'll probably change to where we're actually skipping the animals potentially, which would be very, very nice. The models that we use right now today in 3D actually seem to emulate what the responses would be in humans to a degree. We have to do a better job of validating that, but once we do, I can definitely perceive that we'd be able to get past some of the animal testing.
I think 3D models will be a very important step forward. I think, as we've seen, not all animal models translate to clinical utility in humans. And I think having human animal models as organoids would be an important step. We do use it routinely, but our work primarily involves animal work at this time. But of course, as the technology progresses and more viable organoid options are available for us, for our clinical indications, we'll certainly utilize it.
Yeah, since the Modernization Act passed, I've definitely seen an increased interest in the 3D and especially organoid work to replace animal models. Obviously, they help to solve the ethical concerns of working with animal models, but I think even possibly more importantly, animal models are very expensive and can be quite unreliable, and organoids are much easier to work with and could bring the cost of testing your drug or therapeutic down considerably.